And would you imagine, as we go from our oral cavity all the way down to the distal end of our alimentary canal, what kind of epithelial changes might you expect? Again, here, this kind of brings up the structure governs function kind of relationship. What kind of epithelium would you expect in the oral cavity? Why? She said stratified squamous. How come? She's correct. Would it be wet or keratinized? It would be wet, yes, okay, not keratinized. So why would it be there in the oral cavity? Why not have simple columnar or simple squamous in the oral cavity? What does stratified squamous provide for us? Protection, yep. Would you expect it way down here in the distal end? Change? If you have an opening that is going to be exposed to the air and other caustic chemicals, you would want to have a change in epithelium, and that's what you see. But remember, when you go through most of the alimentary canal, you do find a simple columnar epithelium throughout almost the entire GI tract, with the exception of the oral and the distal rectal anal area down here. But the big thing is, yes, recognize that and appreciate that, but also keep in mind with simple columnar, those cells are notorious, at least the columnar cells are notorious for having microvilli, okay, associated with them. Also notorious for having goblet cells stuck in between them, okay, either in small amounts that you'll find maybe in the duodenum and the jejunum and ileum, but more increasingly in numbers that you'll see in what portion of the alimentary canal especially. Maybe the colon, does that seem reasonable? You find an extensive amount of goblet cells with their mucus. How come?
Oh, that's just an eraser.
What does goblet cells and mucus can be provided in the colon? Lubrication, right? Lubrication. Okay, structure and function, all right? So, that is something you need to be aware of. Now let's go through the slides. Now I'll stick to my slides here. Last time we talked, I gave you a nice overview and pretty much gave you the entire notes in like a 20 or 30 minute presentation, which is okay. Sometimes people like that, sometimes people don't, but sometimes it works out well for overview. So let's stick to the slides here as best we can, and the best I can, right? And so I got some processes here, all of which I've already mentioned, at least in some form or fashion. We have ingestion, taking food in. We know that we're going to take food in. We're going to chew the food up with muscles of mastication. That's the word for chewing, right? Okay. And then we're going to have this bolus of food we want to swallow. And we actually need a word for swallowing. Anybody know? Deglutition. Deglutition is swallowing. Deglutition, all right? And we know that if we're going to swallow something, we like it, the taste buds come into play because we happen to like whatever it is we're chewing on and swallowing. Deglutition is swallowing. So now we have ingested, and our propulsion is this peristaltic type of movement that is moving our bolus of food down the esophagus, and we have, of course, this extensive amount of skeletal and smooth muscle in the esophagus as it moves our food down, right? We get into now mechanical and chemical digestion. That doesn't mean that it is next. It could even be up here in the oral cavity too. So be aware these are processes and they go on all the time, even though they look like they're one after the other. It seems like it could be. But we have mechanical and chemical. When you think of mechanical digestion, what comes to mind? What kind of process here? Yeah, so we got a physical destruction or physical degradation, if you want, of big stuff into smaller stuff. That's mechanical. So there's no chemical process going on here. No enzymes are being used to break anything down. And so we know that's going to be mechanical in the oral cavity especially, but also we'll find mechanical going on in the small intestine when we have bile come along. Where's bile made? Bile is made in the liver and stored in the gallbladder, yes. When we get to bile a little bit later, it's going to be a chemical, or rather a mechanical digestion, not a chemical. Okay, it's actually going to take the big fat globule, as we'll see, and break it into smaller fat globules. That's going to be a mechanical type of digestion, but not a chemical. But eventually we have to come across chemicals that will digest these smaller fat molecules into even tinier structures that will be known as free fatty acids and glycerol molecules. Okay, so we'll have to involve some chemicals there along the way to do that. All right, so, and we go into absorption a little bit later. Where does most absorption take place? In the small intestine. How come? What would you expect? Structure governs function. So what would you expect in the small intestine? Extensive amounts of Microvilli, yes. And he said villi, and he's absolutely correct, because you do find these villi, these upfolds, but also and I zoomed in on this one, you see microvilli. He's absolutely correct by saying villus. I just have to say microvilli because I had kind of zoomed in a little bit more, higher power if you want with the scope, but he's right. We also will be able to see not just the villi and the microvilli for increasing surface area for absorption, but also we'll see that the lining itself, the mucosa itself, is thrown into folds, extra folds. All right, and these folds do not change like you'd appreciate in the stomach. We call those rugae, right? Rugae do change. But when you get into the duodenum and the jejunum, you have these extra folds of mucosa that do not change. They always will be there, no matter how much chyme is in there. They're always going to be there. So that's kind of a difference with looking at the folds of mucosa. You do find the rugae to go kind of flat when there's food in there, but with the extra folds you see in the duodenum and jejunum, they don't change shape, and you actually have a word for that, plica circularis. Extracircular folds is what that means. Plica, P-L-I-C-A, circularis, extracircular folds that you find in the duodenum and the jejunum especially, because again, we can throw this mucosa into extra folds, we can now stack on more villi and then more microvilli, really increasing the surface area for absorption. Okay? So we have lots of ways to do that, one of which again is throwing the mucosa into folds, but also though adding villi and now microvilli. Alright, so that is what we can expect with absorption. Any absorption going on in the oral cavity or in the esophagus or anywhere else. In the oral cavity you might find what being absorbed? In the oral cavity? Yeah, glucose does actually make its way in, absolutely does. What else? Some drugs like medicine like nitroglycerin absorbed sublingually. There are others too, that's the one that comes to mind for me. If you come into a clinical course and the drug is administered sublingually, I just happen to be familiar with the nitroglycerin more so than others, but certainly drugs can be administered sublingually. Alright? Any absorption in the esophagus? Any absorption there? No, there is none, okay? None at all. What would you expect to see in the esophagus as far as the glands? See a lot of glands or little glands, and what would their product be? Yes, what would their product be? You want this esophagus being just dry, so you want a lot of mucus. Yes, mucus glands, right, lots of them. So we make our way down the esophagus. Any absorption in the stomach? Alcohol and aspirin and even water is absorbed in the stomach. It does pass through the lining of the stomach, so or the mucosa if you want. So you do have absorption there. Okay? Alright, defecation is the last one. Just general activities here, essential activities, easily be appreciated, I would suspect. Alright, so we grouped the organs already as far as alimentary canal organs and accessory organs. Everything else is going to be accessory unless it is the conduit, unless it is that tube, it's going to be accessory. And we'll come to those. Any questions so far? Okay. Alright, so 4 basic layers here are the tunics. This is not a big ticket item necessarily to spend a lot of time on, but I just want to address a little bit of it because it actually goes to a certain disorder that is of interest here. We've got layers, mucosa, the lining of course, right, which is simple columnar. Then we have an area known as submucosa and muscularis externa. What would you guess would be in there? At least this one you could probably surmise, right? Smooth muscle, right? And even in here, you're going to find, in addition to this, not just smooth muscle, but also you'll find nerve plexes. Alright, nerve plexes. Then you remember learning about the part of the peripheral nervous system that was called the enteric nervous system, right? Enteric. Did I get to this in your class? I did? Not AP1, but AP2. No, not yet? Okay. Enteric nervous system, okay, is the nervous system that is in the small intestine and is in the colon. Okay? That type within. And it actually functions autonomously. It doesn't need any nerves going to it like the vagus nerve would be doing that. And it does, but if you pull off the vagus nerve, it will still operate. Okay? So we find enteric nerve plexes in these areas, submucosa and muscularis externa, and the serosa is simply the connective tissue covering that covers these organs. And it's not unique to any particular portion of the colon or small intestine, but it's just a connective tissue covering. So what's the big deal here about these two? Why am I talking about this enteric nervous system? Suppose we have a colon like this, okay? That's a bad colon, isn't it? Yeah. Okay. We've got a colon like this, We'll start like this and this and this. There, that's a better colon, okay? And now we notice that we've got some movement of our foodstuffs in the colon, which is essentially waste, right? So it moves and moves and moves and moves and moves like this. Then it comes to an area here where there aren't any more nerve plexes. Maybe a person was born with the lack of nerve plexes that are in the submucosa and the muscularis externa. What do you think would happen when you have this newborn baby and all of a sudden it's eating and so okay, and now all of a sudden it's not eating and starts throwing up and now we've got this area here that starts to go like this, kind of bulges out like this, okay? Kind of looks like a big colon. It's called congenital megacolon. This absence of the what is known as myenteric plexes, congenital megacolon, okay? Absence of the myenteric, which is within the muscle, nervous system in the muscle, right? Which I just said, myenteric plexes in here. So what do you think? What do you think would happen to this person? Yes, you have to go ahead and resect the portion of the bowel that is not working, connected to there, and now things can move along, okay? And this is also has a special name besides congenital megacolon. A person is born with the absence of these plexes in certain parts of the colon, and you end up with another, which is the same name, just a special name, it's an eponym, is Hirschsprung's disease. I always spell this wrong, but you think I'd be able to spell it right because I have this. Hirschsprung's disease. I was born with congenital megacolon, and I'm missing 6 inches of colon. So that's a little extra, but this is not extra in that Hirschsprung's disease, congenital megacolon, is the absence of the myenteric nerve plexes in the colon. Okay? Alright, so that congenital megacolon. That's the important portion about this. Yes, we know it's going to keep our foodstuffs moving along, but sometimes things don't go the right way, and you end up with an absence of the nerve plexes, and up the congenital megacolon, which is Hirschsprung's disease. So in that case, it was resected there and then connected back. So 6 inches in my case is gone. So that's extra. The 6 inches isn't a big, testing question, but nonetheless, that kind of information is of interest. Okay. Any questions on that? So far, so good? Yes.
Why can't the food behind it and the rest of the intestine just keep pushing it kind of through? Why wouldn't the rest of the food just push it?
Because when it gets here, okay, it doesn't, it goes, and then nothing. It doesn't go anywhere else.
But wouldn't the food behind it push it through?
Perhaps, but what if the person says, you know what, it's not going anywhere, I'm not hungry anymore, so now I start to vomit, okay, now it's coming out the anterior here, but this is not going anywhere, and if this stuff doesn't go anywhere, it sets up a nice area, and even a perforated colon could happen too, where you can bust open, but it doesn't move things along because the person will stop eating, but you still have this big mass of material here that doesn't go anywhere. Does that help?
Yeah.
Okay. All right. And that's one of the things to look for in a newborn baby that doesn't eat, is just that. Okay. All right. Or doesn't pass meconium, right? For that. Are you done? That's okay. So, saliva. What do we can say about saliva? Not a big ticket item for us to spend a lot of time on, but what about saliva as far as where it's coming from? Refresh your memory from the other day with the glands that we recognize, like submandibular, sublingual, parotid, yep, and should be it, right? That's not it. That's all, right? There's no other additional ones. I was thinking 2 sublingual, but that's correct. All right. We know the enzyme's in there, which is going to be salivary amylase, right? And we also can find a little bit, but it's not very effective, is lingual lipase, sometimes even called salivary lipase. Not overly effective, but we still have it. It doesn't do a whole lot for us. And I think I've talked about
What was that called again?
lingual lipase. Lingual lipase, we do find it in the saliva, not overly effective. We have to figure out another enzyme that will be able to take over the breakdown of the fat, but we'll have to get to that later today. Okay. All right. A little bit about teeth, not to spend a lot of time on this either, but this is, we have to address a little bit of it just to be dangerous here. And so we look at the teeth. How many teeth are we supposed to have in the adult? Anybody know? 32, yes. Probably have 16 on top, 16 on bottom. And you notice that we've got here, not to identify when they're going to be erupting. That doesn't matter. It's going to have an image that didn't include this. We can recognize we have incisors right in the front. Those are central incisors. And then on either side of those are lateral incisors. And then on the lateral side of those, sometimes called the eye tooth or the canines is what they're called. You can see them positioned right over here is my canine right there. And also, as you go further posterior, you get to molars. There's, let's see, two different names for these. We can call them premolars, or sometimes even people call them bicuspids, which is the group back here. Premolars, or even you can call them bicuspids if you wanted. You go back even further, you get simply the molars. All right. And so what's the big deal about this is just appreciate the shape that these teeth present. With the incisors, central or lateral, they tend to be really sharp. What are they good for? Cutting? Yeah, good for cutting. What about the canines? Tearing, puncturing for sure. And then we have now, of course, our mandible just doesn't do this, does it? Okay, we can tear, we can cut, we can puncture with the canines, but we also though have our mandible that goes up and it goes like this, because when your mandible goes up and down, right, it doesn't just do this. It goes down and then it rotates like this, because we have our pterygoid muscles that move it side to side, right? So we have to take advantage now of the shape of the molars. If we're going to grind something big into something small, that's what the molars do, is grind up this substance, whatever it may be. So we can take a bite of whatever it is, chew it, and then of course grind it with the molars. So now we have our molars back there. We have a name for this one though. The third molar oftentimes, doesn't always, but sometimes they become impacted and doesn't make its way out. That is the wisdom tooth, is what that is. Okay, the third molar, the wisdom tooth. And you're actually welcome to use that term as the wisdom tooth or third molar. I'll accept that. It's pretty popular, so I'll allow that. But what about muscles? And that's what I have. You can cross out the parts and things. I don't spend a lot of time on the parts, just like I don't spend a lot of time on hair and nails and A&P1. That's all fine and dandy, but we got to get some bigger ticket items. So, but we have to at least stress this: the muscles involved in mastication. Refresh your memory. We have the pterygoids, which collectively is a nice name to have, and then we have the masseter and the what? Temporalis, right. What nerve controls those muscles? It is trigeminal, yeah, trigeminal, very good, okay? And then, of course, you can call it the fifth cranial nerve if you want, or the trigeminal, whichever one you wish, all right? This is a good example of mechanical digestion, breaking big stuff down into small stuff. All right, what about the tongue? Also in digestion, so we have to include it, but we have to say, hey, it's not a big ticket item, but it has to be addressed a little bit. All right, so some functions. What is the function of the tongue? Taste, right? That's a big one. What else, though? Moving food around. Moves the bowls of food around. Position your mouth accordingly. So, what about some surface attributes? If you look at your tongue, the dorsal aspect, this is the top of the tongue, right? The dorsal side there. You can see little bumps, which are papillae. All right? And these are all little bumps that you see here. What is in the papillae? Taste buds are sitting in there, right? Taste buds, okay? Taste cells, oftentimes they're called there too, all right? So taste cells. What are our sensations of taste? I think we listed these last time, right? Sweet, the obvious ones, that's probably no surprise to anybody, but sweet, sour, salty, bitter, and umami, U-M-A-M-I, umami, and that's associated, it comes from the Japanese, and it means savory, is what it means. Umami means savory. And it's associated with the amino acid glutamate. That's what it detects, is glutamate, is what that is. So, of all of those, sweet, sour, salty, bitter, and umami, which one of those do you think has the lowest threshold? It is the bitter one. Bitter, poisons tend to be bitter, so that you tend not to eat as much. If it's bitter, you don't eat a poison. That's the idea behind that, but nonetheless. All right, what about nerves that transmit the sensations of taste? Any ideas? What nerves are they going to be? Spinal nerves or cranial nerves? Cranial nerves, right? They have to be. Cranial nerves are the head and neck area. So, what nerves transmit the sensation of taste? This should be a review from A&P1. That's why I have included here.
Says olfactory?
No. I see where you're headed with that. but it's not. It's not. Olfactory is purely smell, but as you imagine, there's some overlap as far as the interpretation of the taste. Facial, yes, facial nerve, absolutely correct. Does a sensation of taste. In fact, most of the tongue, the anterior 2/3 is the facial nerve. What other nerve might be associated with taste? Glossopharyngeal, that's number what? We can go through all 12 if you want. No, 9 it is. 9 it is, glossopharyngeal, okay, is number 9, okay? So we've got that. Be able to recognize at least one cranial nerve that transmits a sensation of taste. Makes for a nice fill in the blank, by the way. So be able to recognize one, okay? I'm sorry? Number 7. Facial nerve is number 7. Okay? Alright, how about motor movement of tongue? Because we're going to move the bolus of food around, we have to have some nerve control over this, and that's going to be cranial nerve number... Yes, hypoglossal, she said that's number 12. Hypoglossal. Okay? Alright. And that's enough on the tongue. Okay? Yes, I enjoy special senses as much as the next person, but we can't spend a lot of time on the transmission of these impulses up to the brain because we have to get into digestion. Nonetheless, here we can appreciate our papillae, where our taste cells are sitting, and as we look here, I've got most of the important information highlighted in red. We can see fungiform and filiform noted here. Fungiform are most abundant right in the dorsal aspect of the tongue, look like little mushrooms, right? And then the filiform, those are papillae that do not have any taste cells in them. Okay, that's the big deal with this. There aren't any taste cells in the filiform. Okay, what are they good for then? Would you think? Friction. They're good for friction, and they're very good for moving the bolus of food around in our mouth. That's what they're associated with. Okay, no, and that's it. No taste buds in there. So circle that one so you don't miss that one. And the other ones are foliate, which are difficult to appreciate, but they're on the lateral sides of the tongue, way on the back. You almost can't see them unless you could pull your tongue out, but you can't because you have these little reins, little frenula, right, that hold your tongue in. You're going to cut it though, like Gene Simmons probably did, right? You can pull it out. But anyway, you got that. And the circumvallate, which are pretty good, pretty gigantic structures, and that really is about the number. They're about 9 to 12 or so. That's only extra because you're probably thinking the artist, why did he hold a whole bunch of them? Okay, they are pretty scanty anyway, about 9 to 11, but those are the circumvallate. And each one of those do possess tasting cells that detect every type of tasting, whether it's sweet, sour, salty, bitter, or umami. And the old saying where you can taste certain substances on certain parts of the tongue more so actually is not the case. It's been shown not to be the case where you taste sour on the sides and bitter in the back. That's not the case. You do taste all of these substances all over the place on the tongue. So that's a little extra, but at least I kind of want to clear that up from what you may have heard. All right, so let's get to our rest of the oral cavity here. Pharynx, we know the pharynx, of course, is what does pharynx mean? Pharynx is throat. It's pharynx, right? And we have different parts to it. We have a naso. This is nasopharynx, as you might suspect, right? Oropharynx, right? And then we go down here to the laryngopharynx, right there. We have our nasopharynx, all this area here. Oro, as we get into here, and then laryngo, as we get down to here. Okay. And so, would you guess that we have different types of epithelium as we go from oro or nasal to oro to laryngo? Yeah, if we're in this area, what would you expect the epithelium to be? Like respiratory epithelium? Yeah? Nobody knows? Yes, that would be the case. You'd find it. As you get into oropharynx, what would you expect the epithelium to be? Would it be respiratory or would it be stratified squamous? Stratified squamous, and also holds true as you get down to the laryngopharynx. Structure governs function for that, okay? And with the pharynx, what's the big deal as far as the muscles go? And that includes part of the esophagus here. What the muscles, they were those constrictor muscles, weren't they? I said that before, constrictor muscles that when they contract, they squeeze the bolus of food down through the laryngopharynx into the esophagus. Constrictor muscles, that makes sense because they're squeezing and moving it down. Alright, and esophagus, we've done histology already as far as the muscle features go, right? Stratified squamous in there, and it is and there's a picture of our stratified squamous and all of that. What do you think these could be right here? No, you don't see them very well, but There's one there, here, here. Those are glands. Those are glands producing mucus. That's what they are. Okay. So far, so good? Histology, all right? All right. Let's do some stomach anatomy here, and then we'll get into histology of the stomach, because we have an interest, of course, of the cells and what their product is, and that's really, really important. So a little bit about the anatomy of the stomach so that we can have an idea of what we're talking about here. Esophagus meets the stomach right there. That's called the cardiac part, cardiac region. And then, and if I'm going fast, that's okay, because we will see this in lab as well. So don't sweat if you're not getting all this right now, but I want to keep giving you an introduction of this, because if I just start talking about this, you wouldn't have an idea of where it comes from. So kind of just sit tight and don't think that we're not going to come back to this, but we are. Okay. So we have the cardiac part. This is the fundus right here. Kind of this upward area here is the fundus. Then we have curvatures. Greater curvature, all that, greater curvature, and lesser curvature there. All right. Now, who went to the cadaver lab? Right. We have this big fatty apron hanging off the greater curvature of the stomach. What was that called? Greater omentum. Very good. Okay, so that hangs off. Do you think there would be an omentum right here? It's called lesser, right? And a lot of you cut through that in order to see the hepatic portal vein in the cat, right? Okay, so there's our curvature. We now also have a constricted portion here called the antrum, right? This is the antrum there. Sometimes even called the pyloric part. You can call it whichever one you want. Antrum or pyloric part is a constricted portion, okay? You also can appreciate here the muscles in the stomach, smooth muscles. They go in all different directions. They're transverse, they're circular, there's longitudinal, okay? And so, of course, what would you say that the function of these muscles would be? Churning, mixing? Yeah, churning and mixing. We're mixing now our bolus of food with the chyme, right? With the gastric juice, which we then call chyme. That's what that is. We've got our bolus of food, we've got gastric juice, mix it together, we have chyme. What's the pH of chyme? Approximately 1 to 2. Very good. So I'll ask that several times as we go through. And we also see the rugae. That's the rugae that you can appreciate there. You'll notice that in your cat. We get the cats back out for one last section. You'll see them there. Okay? All right. So we know the function of the stomach is what? Primarily to do, but it does other things. So what's primarily it's good for? Storage, yes, it's a holding tank for food, but we also know now it's good for mixing our gastric juice with our bolus of food, and we have that called chyme. What about any enzymatic breakdown? Enzymes in here, perhaps? What do you think? That's a big question. Any idea what foodstuffs can be broken down in here? Is it going to be carbohydrate? Is it going to be protein? Or is it going to be fat? Primarily, it's going to be what? Protein. Okay, primarily protein. So we're going to need an enzyme that starts to break down protein, because in the oral cavity, nothing happened. When I told you, hey, you put a peanut in your mouth, what is it? Still a peanut after 5 minutes, right? Nothing happened, okay? That wasn't the case with carbohydrate, and it was pretty much the case with fat, but there are some changes because of that lingual lipase. But we go to protein now. This is one of the major functions of the stomach, besides being a holding tank, is the function as an area where protein breakdown chemically starts. So we need now an enzyme that starts to break down proteins, and it's called pepsin. Okay, pepsin. We don't know yet where it's coming from, but we'll get to that, right? And how it's made. But pepsin, okay, is a chemical that can be used for protein breakdown, cleaving proteins into what? Dipeptides, tripeptides, and even amino acids in some cases, okay? But that's where protein is going to start, with pepsin, okay? All right. You think there's also going to be a little bit of this? Maybe you do or don't. Gastric lipase. There's a little bit of that as well. Not overly effective. It's more effective rather than what you see in the oral cavity, but it definitely needs some work, because that's not very effective, at least in the stomach. We have to have another lipase coming along. Any guesses where another lipase will come along as we continue on here? Where could another lipase come from? Very, very important organ. Posterior to the stomach. It is the pancreas. Yes, and you might even call it what? What lipase? Pancreatic lipase. Yes, very, very important. All right, so we've got the function, the parts at least identified there, and so we can now look from microscopic presentation here. This is a light microscopic view, and we can see that the stomach has in it what are known as gastric pits. All these are pits, okay? We have the mucosa thrown into folds, but if you zoom in at a higher power, you'll see we have pits here, okay? Gastric pits. And all of these pits are formed by these structures here, which are gastric glands. So as you descend this gastric gland, you're in a gastric pit. And of course, we're going to find various cell types in there. And we're going to figure out what these cells are and what their product is too, okay? All right. And there's our parts there of our structures that you already know. Pyloric part or antrum, whichever one you wish. All right, so the histology of the stomach, we have an idea. Gastric pits is what we know already, and we have our cells here. So if we look at a gastric gland, right at the top, you find this mucosa, or rather mucus cells that produce mucus. It's not as viscous as the mucus that you're going to find in the GI tract, but in the small intestine and colon. But it is mucus that does seem to protect the lining of the stomach, although it's not overly viscous, but it still does help protect the lining of the stomach, mucosa, from this HCl, right? Okay, probably no surprise. Parietal cells, they have several functions. Parietal cells, you will not have to identify parietal cells histologically, okay? Don't worry about that. But you have to be aware of what the products are. One of which is what? HCl, hydrochloric acid. Okay, HCl. Would you think that this cell is going to be able to produce HCl within its cytoplasm and have it at the ready and say, hey, I'm going to be told to secrete HCl by some other chemicals that I'm coming to, and when it gets to me, I'm going to secrete HCl? You think that's a good idea for that cell to have HCl kind of pre-packaged inside of it, or you think that would be very bad if you could have that? Actually, that's very bad. A cell is not going to have HCl, okay, within its cytoplasm, right? As we'll see in maybe tomorrow, not maybe we'll see tomorrow, we'll come across something known as a proton pump, okay, which will be pumping out hydrogen ion into the lumen of our gastric gland, and we'll also find in there in the lumen is chloride, and then those joined together will make our HCl, all right? So just as an aside here, actually it's not an aside, okay? Would you also think that if this cell is producing HCl, pumping out hydrogen ion, okay? Think of it as producing HCl, but it doesn't secrete as a whole, okay? It pumps out hydrogen into the lumen. Would you think that if it's doing this, and maybe someone has maybe some of this gastric esophageal reflex disease, GERD, maybe some heartburn if you want, okay? We can call it that if we want to. Would you think one of the treatments could be, hey, why don't you take a drug that blocks this proton pump so that it pumps out less hydrogen ion, forming less HCl? See, reasonable? There is a drug, okay? Prilosec does that. That's a proton pump inhibitor is what that does. So that's one treatment for gastroesophageal reflux disease, or GERD, heartburn if you want. That's one treatment for that. Prilosec, P for Prilosec, P for pump. Okay? Alright, so we got parietal cells doing that. We also have parietal cells secreting this intrinsic factor. Intrinsic factor. They make intrinsic factor. Okay? Parietal cells do. This is a molecule that binds vitamin B12, okay, and transports to the distal ileum so that we can absorb vitamin B12 that way. All right? Vitamin B12 is coupled to intrinsic factor, and when it gets to the distal ileum, it actually protects it from the acid in the stomach, and it makes its way to the distal ileum where we absorb vitamin B12. That's the only place we absorb vitamin B12 is in the distal ileum, coupled with intrinsic factor. All right? And so we absolutely have to have intrinsic factor. You can't live without intrinsic factor because you can't absorb vitamin B12 without it. So we have to have vitamin B12. And you know, the stomach just is a holding tank, and that's great, but it really is, this is its only vital function, is producing intrinsic factor. Without it, you can't absorb that. Without that, you can't make, well, many things, but one of which is red blood cells you couldn't make, okay? You end up with anemia, okay? And there are different types of anemia in this particular case, which is called pernicious anemia in that case. But nonetheless, anemia. So this is the only vital function of the stomach is to make intrinsic factor. That's it. So you could live without a large portion of your stomach, but if you took out the portion that makes this, okay? Because not every area does everything, okay? If you took that out, you would have big trouble. Okay? That is the major function of the vital function of the stomach is producing that, so we can absorb vitamin B12 in the distal ileum. All right, we've got parietal cells done. Now we need these, chief cells. Chief cells, all right? They produce something known as pepsinogen. Pepsinogen, okay? That's what they produce. Would you guess that pepsinogen is going to be cleaved and converted into pepsin? That is correct, pepsin, okay? Pepsinogen. Now you're probably saying, well, what will cleave it and convert it into the inactive, or the active pepsin from the inactive pepsinogen? And that will be HCl. We'll do just that. We'll cleave pepsinogen into pepsin. That's what HCl is going to do. Okay? Alright. We also have gastric lipase out of these cells too. Alright, I won't say much about that, but that is another function of the chief cells is gastric lipase. Okay. Now we need the last one there, which are kind of a weird name, but enteroendocrine cells. Enteroendocrine cells. What do you think this would be associated with? Entero is within. Endocrine would be what? Some type of hormonal type of cell? Yeah, hormone producing. So I've listed a bunch of them, and I've kind of put up this to jog my memory there. Enteroendocrine cells, and they're way at the base of the gastric pits. And the first G there is gastrin. Gastrin is a hormone that's produced by these enteroendocrine cells. Okay? And the first thing that gastrin does, or I should say the first thing that causes gastrin secretion is the presence of food in the stomach. That stimulates gastrin secretion. Okay? So we got to figure out when this comes about. As soon as you get food in the stomach, gastrin is produced. Gastrin then does a couple of things, one of which is causes the relaxation of the ileocecal valve. Ileocecal valve. That's what it does. Causes relaxation of the ileocecal valve. So as soon as you get food in the stomach, it says, hey, there's food in the stomach, let's move everything down the line. Okay? That's what it does. Right? It also increases HCl production. So what would be gastrin's target then? If the hormone's got to go somewhere, where would it go to? What cells? Parietal cells, absolutely right. So we got relaxated ileocecal valve, increased HCl production. It also causes mass movements of the colon. Mass movements of the colon. And think of a mass movement of the colon as not just kind of peristalsis in one location, but an entire area of the colon all contracting at the same time. Doesn't have to be entire transverse colon, okay? But certain areas all at once, just contracting all at once and just big mass movements. That's what it does. And so, as soon as you get food in there, these things happen. So maybe you're saying, well, give me an example. Perhaps if you are maybe, as soon as you, well, I don't need the example, I guess. We get the idea, right? As soon as food gets in the stomach, it feels like maybe that you maybe have to move your bowels or something, right? Maybe if that's the case. But that's maybe an example that could come to mind, all right? So we have ghrelin now. Ghrelin. Okay, this is an unusual one. Ghrelin does come from these cells here, and I have in parentheses another one that is not associated with the gastric mucosa at all, but they go together. Ghrelin is a hormone that stimulates your hunger, okay, hunger center. And where would that be? In the hypothalamus, yes. So ghrelin is activated when there's not any food in the stomach, okay, when you are hungry. Ghrelin is released. Goes up the hypothalamus and stimulates your appetite. That's what it does. Ghrelin. Okay? But we also know if we've got a hormone doing this, whatever this is, there's always going to be another hormone doing the opposite, whatever this is. So in this case, we have